I am Nagaiah, Professor in Biology. I am having 35 years of teaching experience. Especially, I am handling the NEET classes. In this academy, means Excellence in NEET Academy, I am specifically handling the NEET classes for so many years. And this is the academy, famous academy in Bangalore. It is present in Vidyaranapuram. Today, I am going to discuss about the second PU genetics chapter. To start with, let me discuss about the basics of the genetics. Okay? Genetics is a branch of biology. It deals with the studying of the heredity and variation. Heredity, the transferring of the genetical characters from generation to generation can be called as a heredity. Variation, it's a differences between the parents and the children or among the children of the same parents. So that studying of both the heredity and variation constitute the genetics. And one more definition can be given. Simply, genetics means the studying of genes, right? Gregor Mendel, also called as the father of modern genetics, he laid down the foundation stone to this genetics chapter. He conducted the experiments, genetical experiments on garden pea plant. The scientific name of this plant is Pisum sativum. Mendel conducted the experiment on this plant during the period, it is 1856 to 63. Okay. That Mendel studied totally 7 characters in this plant. We can say 7 characters, or you can say 7 pairs of contrasting characters, right? The important characters he studied were the plant height, the first character he studied. Then he studied the color of the seed, okay, shape of the seed, color of the pod, shape of the pod, like that, color of the flower and position of the flower. These are the 7 characters studied by that Gregor Mendel, right? Then after studying all these characters, he formulated the laws. They were famous. They were popularly called as the Mendel's laws. Okay. So in this chapter, I am going to discuss about the monohybrid cross experiment and the dihybrid cross experiment and incomplete dominance. Like that, there are so many topics are there. I will discuss that later. To start with, before studying the actual topics, let us discuss about some definitions concerned with this chapter. For example, hybrid. Hybrid, it's an individual. It is obtained by crossing 2 parents which are differed at least in 1 pair of characters. Okay, this is hybrid. In this hybrid, it may be monohybrid, it may be dihybrid, like that. Monohybrid, the hybrid which is obtained with respect to 1 character can be called as the monohybrid, like that. The hybrid which is obtained with respect to 2 characters can be called as the dihybrid. The cross made to get a monohybrid can be called as the monohybrid cross, like that. The cross made to get a dihybrid can be called as the dihybrid cross. Okay. So in this chapter, actually principles of inheritance is the chapter to start with. Okay. So in this principles of inheritance chapters, you will study monohybrid cross experiment, dihybrid cross experiment, but the name is 1 gene inheritance experiment and 2 genes inheritance experiments. Okay. I will discuss that later. Concerned with other definitions coming in this chapter, phenotype, genotype, Let me explain this. Phenotype, the physical expression or physical appearance of an organism is called phenotype. Like that, genotype, the genetical constitution, or you can say genetical makeup of an organism can be called as the genotype. So these are the terms coming again and again in this principles of inheritance chapter. Like that, there is dominance, there is recessive. Dominance or dominant, the character, the allele which is expressed both in the homozygous and heterozygous conditions can be called as the dominant character or a dominant allele. Okay. Recessive, the allele, the character which is expressed in the homozygous condition only, and it is not usually expressed in heterozygous condition. Such character, such allele can be called as the recessive allele. Okay. Like that, then what is the meaning of alleles? You see, before that, I hope you know about the nucleic acids. Both DNA and RNA can be called as the nucleic acids. DNA, deoxyribonucleic acid. DNA is made of many nucleotides, and that DNA having so many segments, the segment of DNA can be called as the gene. So that gene is a segment of DNA, and that gene carries a specific character from generation to generation, right? There are so many definitions are there. I will tell you that later. Come to this point. Actually, each character is carried over by a pair of alleles. Then what are alleles? Alternative forms of the same gene can be called as the alleles. Alternative forms. That gene, if it is present in two forms, that two forms can be called as the alleles, right? If that gene is present in more than two forms, all that forms can be called as the multiple alleles, right? So you see, these are about the alleles. Then you know about the homozygous, heterozygous. Then what is homozygous? If the character is represented by a pair of similar alleles, then that can be called as the homozygous. Like that, if the character is represented by a pair of dissimilar alleles, then it can be called as the heterozygous. Like that, there are so many definitions are there. I will tell you that later. Right, now come to the monohybrid cross experiment in detail. Okay, so as I told you, when the question is asked for 5 marks, explain the one gene inheritance experiment. What you have to do is, first you have to define the term monohybrid cross. As I told you, monohybrid cross, it is a cross made to get a monohybrid, is called monohybrid cross. This is the first line what you have to write. Then actually Mendel what made is, Mendel crossed the homozygous tall plant with the homozygous dwarf plant, got the F1 generation, or you can say first filial generation, right? Before that, I want to say something about the dominant and recessive once again. The character, as I told you, which is expressed both in the homozygous and heterozygous conditions can be called as the dominant. The character which is expressed in the homozygous condition only but not in the heterozygous condition can be called as the recessive character, right? So when Mendel made the cross between the homozygous tall and homozygous dwarf, he got the F1 generation. In F1 generation, he got the tall plants only, but they are hybrid. Hybrid. You know what is hybrid? Hybrid is an individual. It is obtained by crossing two parents, okay, which are differed at least in one pair of characters. Then in the next experiment, Mendel made the self cross among the F1 progeny and got the F2 progeny. In F2, he got both tall and dwarf plants in the ratio 3:1. So this is the explanation you have to write in the examination when the question is asked. Explain the one gene inheritance experiment. After that, what you have to do is you have to write the chart. Okay. For example, parents homozygous tall. Actually, homozygous tall is represented by the capital letters like this. Like that, homozygous dwarf represented by these two letters. So Mendel taken the capital letters to symbolize the dominant characters and the corresponding small letters to symbolize the recessive characters. So this is a homozygous tall plant. This is the homozygous dwarf plant. You have to write here homozygous tall plant. This is a homozygous dwarf plant. Right. So what we have to do is we have to take the gametes from this. When this plant produces the gametes, gametes will be like this. These are the gametes. Like that, this one also gives the gametes like this. When we cross these, you see, cross these two. This is the tall plant. This is the tall plant. So this is F1 generation. This is F1 generation. So as I told you, in F1 generation, all the plants are tall but hybrids. Right. When this F1 is self-crossed, he got the F2 generation. I told you in F2 generation, he got both tall and dwarf plants in the ratio 3:1. Right. Again, to show that, you have to take the gametes once again. This heterozygous tall plant can produce two types of gametes. Gamete with a tall allele, gamete with a dwarf allele. Like this. Okay. Cross these two. What we have to do is, we have to cross both of these gametes with both of them. Okay. So cross these two. This is the homozygous tall. This is the heterozygous tall. Again, this is the heterozygous tall and this is the homozygous dwarf. So you see here, this is the F2 generation. In this F2 generation, we got 3 tall plants and 1 dwarf plant. Okay. So we have to write this flow chart. After that, what we have to do is, you have to explain the same with the help of the Punnett checkerboard. Okay. I will show you that. This is the Punnett checkerboard. Right. This is the Punnett checkerboard. We have to take the same gametes. This is the gametes of one plant. This is the gamete of another plant. Cross this. Homozygous tall. Heterozygous tall. Again, heterozygous tall. Homozygous dwarf. So this is the Punnett checkerboard. At last, you have to write both the phenotypic ratio and the genotypic ratio. The phenotypic ratio of monohybrid cross experiment is 3:1. Like that, the genotypic ratio is, you see here, only one homozygous tall plant is there. Homozygous plant is 1. These two are the heterozygous. 2. This is homozygous dwarf. So the genotypic ratio is 1:2:1. So up to here, you have to write, if the question is asked for 5 marks, explain one gene inheritance experiment. Okay. So I will summarize once again. If the question is asked so, first of all, you have to write the definition of monohybrid cross experiment. Okay. Monohybrid specifically. Okay. I will repeat that once again. The hybrid obtained with respect to one character is called monohybrid. Okay. Then you have to explain the things in 2 paras. Then you have to write the flow chart. Then ultimately checkerboard. Finally, you have to write the phenotypic ratio 3:1 and the genotypic ratio 1:2:1. So this is the genotype 1 gene inheritance experiment conducted by that Gregor Mendel. Okay. Then concerned with that, you might have known the test cross ratio. Test cross ratio. Then what is the test cross? There are two terms. One is the test cross and one more is the back cross. Back cross. Then what is back cross? What is the test cross? The crossing of F1 progeny, particularly with the double recessive parent, is called test cross. I will repeat it once again. The crossing of the F1 progeny, for example, this is F1 progeny. This is the double recessive parent. Crossing of this F1 progeny with this double recessive parent is called test cross. Right? Crossing of the F1 progeny with both the parents, can be called as the back cross. Right? So crossing of F1 progeny with both the parents can be called as the back cross, and crossing of the F1 progeny particularly with the double recessive parent is called as the test cross. Right? What is the purpose of conducting a test cross? Significance of test cross. Test cross is conducted to know the genotype of the F1 progeny, or to know the genotype of the, or you can say, unknown tall plant. Okay, this is the significance of test cross. Then what is the test cross ratio of this one gene inheritance experiment? You see, we have to, these are the gametes of the F1 progeny. This is the gamete of the double recessive parent. Cross these two. You see, this is the tall plant, this is the dwarf plant. Ratio 1:1. So 1:1 is the test cross ratio of the monohybrid cross. Right? So if you write this much, you will get full marks. So based on this one gene experiment, there are so many CET and NEET questions are there. For example, test cross ratio of the one gene inheritance experiment is, there will be 4 answers. Out of that, 1:1 is the correct answer. Like that. The phenotypic ratio of the monohybrid cross experiment, or it may be one gene inheritance experiment, there are 4 answers. Out of that, this is the correct answer. 3:1 is the phenotypic ratio of the one gene inheritance experiment. Like that, the phenotypic, this is a phenotypic ratio. The genotypic ratio of the one gene inheritance experiment is, there are 4 answers. 1:2:1 is the genotypic ratio of the monohybrid cross experiment or one gene inheritance experiment. Okay. So like that, there are so many questions are there. I will tell you that later. Then, as I told you, Mendel conducted the dihybrid cross experiment also. After conducting the monohybrid cross experiment, Mendel formulated 2 laws. They are popularly called as the Mendel's laws or principles of Mendel's. Okay, that 2 Mendel's laws are, one is the law of dominance. Law of dominance. And the second one is the law of segregation. Law of segregation. These are the 2 laws proposed by the Mendel after completion of the, after conducting the monohybrid cross experiment. Okay. Law of dominance. That law of dominance is also called as the principles of dominance. Then what is that law of dominance? According to law of dominance or principles of dominance, okay, I will tell you the definition. If 2 contrasting alleles or contrasting characters are brought together in F1 generation, only one is expressed, it is called dominant. Other is suppressed, it is called recessive. This is the law of dominance. I will repeat that once again. If two contrasting characters or contrasting alleles are brought together in F1 generation, only one is expressed, it is called dominant. Other is suppressed, it is called recessive. This is the law of dominance. Right. For example, you see, this is the tallness allele, this is the dwarfness allele. When these two are brought together in F1 generation, you see, this plant will become tall plant only because this allele is a dominant allele. This is recessive allele. That's why this plant becomes tall plant. This is the law of dominance. Okay. One more definition I will give you. Simplest definition. Expression of the dominant allele or dominant character in F1 generation is the law of dominance. This is the simplest definition for law of dominance. Right. Then law of segregation. Actually, after conducting the monohybrid cross experiment, Mendel explained so many new things, and that new things is introduced, is inserted under the law of dominance in your NCERT textbook. The first point, what you have to write according to NCERT, what you have to write about the law of dominance, according to law of dominance or according to Mendel. Number 1, each character is carried over by a pair of, each character is carried over by a pair of discrete units called factors according to Mendel. This is the first point discovered by Mendel. So the first point is characters are carried over by discrete units called as the factors. First point. Second point, each character is carried over by a pair of alleles. Third point, when two contrasting alleles are brought together in F1 generation, only one is expressed. It is called dominant, other is suppressed. These are the points given in your NCERT textbook under this law of dominance. Okay. So I will repeat it once again. According to Mendel, according to NCERT books given, the characters are carried over by discrete units called factors. Second point, a pair of factors each character is carried over by a pair of factors. Third point, when two contrasting factors present together, only one is expressed, it is called dominant, other is suppressed, it is called recessive. This is the law of dominance. Okay. Then law of segregation. Law of segregation is the continuation of the law of dominance. What it explains that when two contrasting alleles brought together in F1 generation, they do not blend. Instead, they lie side by side, and during gamete formation, each character or each allele is segregated from each other and moves into separate gamete like this. This is the law of segregation. Simply we can say that the segregation of the contrasting alleles or contrasting characters during gamete formation is called law of segregation. Like this, you see, during gamete formation, these two alleles are separated or segregated from each other and moves into separate gametes. This is the law of segregation. So this is a brief information about the one gene inheritance experiment, about the test cross, and about the law of dominance and the law of segregation. Both of them are concerned with the one gene inheritance experiment.